Buenos días a todas y a todos. Nos vamos a dar el gran gusto hoy de tener a cargo de esta presentación, en el marco de los seminarios institucionales del IIBCE, a Andrew Mason, el doctor Andrew Mason. Andrew es profesor en el Departamento de Ciencias Biológicas de la Universidad de Toronto, en Canadá, donde tiene su laboratorio. Hace muchos años que se viene especializando en lo que es la comunicación acústica y comunicación vibracional en su conjunto, en diferentes grupos de artrópodos, como grillos, saltamontes, moscas, y más recientemente, pero ya hace varios años, también en arañas. Nosotros estamos teniendo el gran placer de tener a Andrew aquí en una estadía de investigación en el instituto, y particularmente en nuestro departamento, en el Departamento de Ecología y Biología Evolutiva. Y bueno, hoy Andrew nos va a contar todo lo que ha venido aportando él y su grupo de trabajo a la comunidad en cuanto a la generación de conocimiento de comunicación vibracional. En arañas, creo que particularmente más que nada ahora en arañas de tela, y él sabe muchísimo también particularmente de las arañas de la familia Theridiidae, que son las arañas esas culonas, las que le llamamos las viudas negras, o las arañas del lino acá, y también las falsas viudas negras. Así que es una alegría enorme, Andrew, para nosotros, y un honor tenerte acá, si es que sobrevive a las conexiones. Are you ready?
Yes.
Thank you very much for being here, and go ahead.
Okay, well, thank you very much for hosting me here. It's really a great pleasure to be here. And I hope I can make some sense. And I'm very conscious of the fact that you're graciously allowing me to speak in a foreign language, so I'll try to be as clear as possible and not sometimes, if I think I'm running out of time, I'll try not to. Mike?
Arriba.
Sound again? It's good again? Hello? 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 Okay. We're back. Hey. Yeah? Hello, hello. Pa, 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 pa.
Hola, hola, hola. Este está bien.
Okay. If this one works, I'll stick with that. Is this okay? And I have the handheld as backup. Maybe it has something to do with holding this in my hand. Okay, thank you. As I said, I'm very aware that I'm speaking a foreign language, and I'll try to be as clear as possible. And so I'm going to talk about mainly about some of my work on vibrational communication in spiders, specifically spiders that live in what we call cob-webs. So this irregular, tangled, interconnected, highly interconnected silk structure. But I thought I might start with a very, very brief summary of how I came to this kind of work. I know that Maka summarized a little bit of that during the introduction, so I won't say a lot about that. And then I'm going to talk about spiders and webs and vibration, mainly two species, well, almost entirely two species. The Western black widow and the noble false widow spider. Okay, so my interests, broadly speaking, are in natural sounds. I started out with I don't know, just an innate interest in sounds that I heard in nature, frogs and birds, and singing insects. And I also am very interested generally in animal behavior in the sense of how animals make decisions about behavioral options and how these are adaptive. And this led me to study communication because it's a nice framework in which you have a signal that carries some kind of information that an animal can use to decide between different behavioral options. So I'm also interested in how these things happen, the sensory mechanisms of processing information from communication systems. So I guess the category I might identify with is neuroethology. So that's the neural basis of behavior. Okay, so I started out with singing insects, and that included these. Okay. These kind of crazy, aggressive crickets that are found in the mountains of Western North America. So that's a video clip of these two animals fighting. They have these contests where they sing. They basically scream at one another for a long time, and then sometimes they resort to violence. This is a video of the wing movements of a cricket producing sound. So this is the mechanism of stridulation. They rub their wings together to produce sound. So singing insects led me to work on acoustic parasitoid flies. So these are flies that reproduce by the females depositing parasitic larvae on crickets. And so the point of this clip is that these animals find the crickets by sound. So it's not exactly communication, but they have to detect and recognize the sound of a cricket and localize the source. And so that's an interesting kind of sensory problem for these small flies. So that was mainly a sensory project on directional hearing and sound localization. And then I also started to work on spiders. I had some collaborations with spider biologists. And so this is actually a clip of a Western black widow pair. The female is much larger, and the male is on the web making vibrations by tugging on the strands of the web. And the main motivation, aside from is that the signaling behavior and the communication in spiders is surprisingly complex. So this became the entry for me to work in the larger area of signal complexity and the evolution of more complex communication signals. So how to do that? And I'm going to maybe indulge myself a little bit here, but another interest of mine is, as Macarena already knows, is collecting vinyl records. So I like records, and I have a turntable, and I play records. As you may know, the way this technology works is you start with a recording of sound. So that is a conversion of the pattern of sound vibrations in the air into an electrical signal. And then that electrical signal is copied into a groove on the surface of the record. So the groove actually is a physical reproduction of the waveform of the sound. And then when you want to listen to the music, you place this little mechanical device onto the record that has a small needle that fits into the groove. And then as the record turns, the variation in that groove that copies the pattern of the sound wave induces movement in the needle, and that in turn generates an electric current that reproduces the sound waveform. So you connect that to stereo, you have the waveform again, and it's reconverted into sound, and once again, it's music. So now if you think about, instead of the groove, imagine that as a strand of silk in a spider web, and instead of the needle, we have a laser beam that's directed at the silk on the web, so that when the silk vibrates, that movement perturbs the reflected laser beam, and that disturbance of the beam is a copy of the movement of the silk. So this is the method we use for recording vibration from very, very small surfaces and objects like a spider web. It requires no interference with the surface because it's just a laser beam. And then what you end up with is the vibration of the spider, and we can connect this to an audio system in the same way we would a sound recording and play it back as sound that we can hear, but it's actually the pattern of vibration that the spider perceives through mechanoreceptors, and it's primarily in its legs. Okay, so as I said, vibratory communication is very complex in spiders and very diverse. So this is just a quick example. This is some species, some North American species of wolf spider genus, and the traces show the overall amplitude pattern of the vibrations produced by these different species. So across closely related species, you get this highly variable patterning of communication signals. But as I'm going to describe through the talk, there are themes and patterns in the organization of these signals and the ways that they're produced. So that's something that's of interest. So widespread use of vibration and highly diverse, and in fact, this is a project that I worked on some years ago. This was one of the first spider projects I worked on. These jumping spiders have some of the most complex signals of any invertebrate animals, and I would say they're comparable to the kind of the birds of paradise of invertebrates. And I won't try to explain all of this, but the point here is that the lower figure represents an attempt to summarize the entire display for one of these species, and the two sections represent these animals combine visual movement displays with substrate-borne vibration displays, and so this is a trace of the visual movement. And this is a kind of a notation that summarizes the different elements of vibration and how they're organized. They go through different stages where they add new elements as they progress through this incredibly complicated display. So this is really fascinating. But I guess the point I'm making here is that this is a high level of complexity where the challenge is just to describe and define what the signal characteristics are. So sometimes it's useful to start with something a little bit simpler. And oh yes, so because the complex communication signals are widespread, there are a number of taxa that produce signals that incorporate what seems to be more information than is necessary for the basic information as we understand it. And so it's an interesting problem. Why do signals become more complicated through evolutionary time? And maybe a useful way to approach that question is to look at something that's complicated but not too complicated to really understand. That's kind of the strategy. So, having studied these incredibly elaborate signals in jumping spiders, I moved to Theridiidae spiders, which are these cob-web dwelling spiders. And the signals in this group are less complex than the ones I just showed you, but they do nevertheless include long sequences that incorporate multiple signals that are delivered in different order in an extended display. So there's something to understand there in the context of complexity. And it's also the case that surprisingly little is known about vibration in spider webs, especially these tangled cobwebs. So that brings us to the main topic, vibratory communication in cobweb dwelling spiders. So as I mentioned, you might consider it a lower level of complexity, but that can be a good way to move forward. It's also an advantage that we can rear these animals in the lab, so we can do experiments more consistently. So I'm going to talk about the Western black widows, which is a more or less complete story, and the false widows, which is newer work that's less complete. It's more work in progress. And if I have time, I actually don't know what my time is now because when did I start? Anyway, if I have time, I'll throw in some highlights of some of the recordings we've been doing here. Okay, so the Black Widow project, this was a PhD project of a former student, Sen Sivalingam. These spiders are widely distributed in North America. They have a very striking size dimorphism. The males are much smaller than the females. They build these cobwebs that they get bigger and bigger over time. And when the males are mature, they abandon their webs and head out walking through the environment searching for females. Their initial location of females is based on chemical cues, so the webs effectively have an odor for the males, and they can find their way to a female's web. But once they're on the web, then it becomes primarily a vibrational interaction. So the males find a web, and they start to do this courtship display where they vibrate or they engage in behaviors that look like they would cause vibrations for quite a long time, a period of 1 to 5 hours. And there have been descriptions of the behaviors in the past in the literature, describing things like body shakes, abdominal vibrations, web shaking, and push-ups. So I'll define those things a little bit more precisely in the next few slides. So, but it was when we started this, it wasn't really definitely known how many of these behaviors were distinct signals or generated by vibrations that were different from one another, or versus observations of different subjective descriptions of the same thing. And I should also say that sexual cannibalism is rare in this species. It really only happens if the females are very hungry, and both males and females can mate multiple times. So the main questions for this part were, what are the signals? What information do they carry? And how, do they, and how do they affect mating outcomes? Okay, so this is a trace of about 20 minutes of courtship no, it's longer than 20 minutes. Hours of courtship in a pair of these spiders. So that's just a long oscillogram of the vibration produced by the male and the female if she was moving on the web. And you can identify broadly different phases of the behavior if you're watching the males do this. Initially, the male is moving on the web without being close to the female and generating vibrations. And they're also, I don't know whether I've got, okay, yes. So what we call the distal phase is the male's initial movement on the web before he actually makes contact with the female. After the first time the male touches the female, he enters what we call the proximal phase of courtship, where the pattern of signaling is slightly different, and I'll describe that. And then finally, there's the copulation phase, which is, in these animals, is relatively brief compared with the overall duration of courtship. So, sorry, I didn't. Is there a way to make that a little bit louder? It'll be more fun later if we have it slightly louder. So that's a male in the early stages of courtship, and one of the other things they do, as you can see here, is they cut the web and bundle it up, so they reduce the silk in the web. And that's believed to be a way of reducing the odor that might attract competitors to the web. So sometimes it doesn't work out, but that's a male. In the proximal phase, several things are happening there. I'll show them in more detail. So he's doing several things. The abdomen is vibrating, the legs are moving against the web, and sometimes the whole body is moving. And so we recognized that there were these at least three distinct types of vibration. So abdominal tremulation, if you watch the videos closely, you'll see that the male's abdomen is moving up and down. So the upper traces here are the amplitude patterns for these vibrations, and then the lower traces are the frequency spectra representing the relative energy at different frequencies of these signals on the web. And I have to say that there are some differences here, but there's a severe constraint of the mechanical properties of the web. So there's a limit to what the male can do to make the web vibrate at different frequencies. But the abdominal vibrations are low frequency, and low amplitude. And then the bounce signals. So this is where, I hope this is a good one. Higher amplitude, this is the male basically slamming his body against the web. And the web plucks you've seen in some of the other videos, those are leg movements on single strands of the web. So there's some broad frequency differences here, and there's certainly big differences in the amplitude patterns, especially for the abdominal tremulation. So the next thing we did was to look more precisely at the body movements that are associated with that body movements that are occurring during the production of these signals. And the diagrams here represent so using high-speed videos, we trace the movements of different parts of the body, and the black traces are the vibrational waveform, and then the traces below that are the movement patterns coinciding with that vibration for the different body parts. So there's a one-to-one relationship between abdominal oscillations and vibration of the web when the males are doing the abdominal tremulations. So he's just moving his abdomen up and down, and the pattern of movement of the web that causes is at the same frequency as the abdominal movement. So there's no evidence of any frequency multiplication mechanism. There's no stridulation involved here. This is just tremulation. Whereas if you look at web plucking, the only significant movement that's happening here is the leg that he's reaching out and grasping a strand of the web and pulling on it. And then if you look at the body tremulation, so there's a retraction of all legs simultaneously so that there's kind of a trampoline effect of the web, and the male's body strikes the web. Let's see. And they also do this when they're on the female's abdomen. Okay, so three distinct patterns of movement, each generating vibration with different characteristics. And you can statistically differentiate these signals based mainly on the power and duration of the vibrations produced. So several distinct signal components are produced. And at least some of these are correlated, have parameters that are correlated with male size. So the peak power of both the bouncing and the plucking signals increases with male size. So there is some information about male phenotype available in these signals. But more interesting perhaps than that was the observation that the sequencing or the temporal patterning of these signal components changes through the course of the overall courtship sequence. Oops. So they produce, as the clips have shown, you see that they kind of seem to be haphazard. The males are moving around on the web, the abdomen's vibrating a lot of the time. They'll pluck the web, they'll bang, they'll bounce against the web. And that seems to be somewhat haphazard. But, so, in the distal phase, this is just a sample of a smaller sample of the sequence. And the symbols above, the red, green, and yellow symbols represent each of the three signaling types, the abdominal tremulation, bounce, and web plucks. And so this is kind of consistent with what your impression probably was, a haphazard sequence. There's no obvious patterning to that. But as you move into the proximal phase of courtship after the male contacts the female, so this is another subsample of the trace with the different signals designated in the same way, and it appears to be much more orderly. So now there's a abdominal tremulation, followed by bounce, followed by a series of web plucks, and that cycles over and over again. So this suggests that there's a kind of different level of organization later in courtship after the male has contacted the female. So it's not a wholesale conversion. They're not always doing this, but they go through cycles of doing this ordered sequence later in courtship. So that is what we would call a rudimentary level of complex multi-component display, a complex signal. And here, I think, is an example. So he's very enthusiastic. Okay. So 2 cycles of that structured display. Okay, so that's interesting. Does it can we show any effect of the organization of the signal in this way? So the way we looked at this was considering only the proximal phase of courtship. We defined what we call the structured signaling rate. So this is counting the number of times the signals are delivered in this sequence and dividing by the duration of that part of the courtship. And looking at that over the whole courtship sequence, it does happen much more. It happens significantly more often in the proximal phase and also in the copulation phase of courtship. So males are doing this significantly more so after contact with the female. So the next question was whether this is simply a gradual acceleration of this behavior or whether it's a transition to different mode of signaling after they make contact with the female. So to ask this question, we just took the proximal phase of courtship and divided it into quarters. So the 4 equally equal durations relative to the total duration of this part of the signal, and then measured the structured signaling rate within each quarter of the proximal phase of courtship, and asked whether it was correlated with latency to mating success or male weight, which was a correlate with some of the other individual signal qualities. And we did this with a manipulation. So the green blob represents a blob of wax that we applied to the male to basically glue the abdomen to the cephalothorax and inhibit them from moving the abdomen. And so we made all these measurements and counted up the signals and compared the rates under these 2 conditions. And it turns out that they do not continually increase their rate of structured signaling through courtship. They switch from distal to proximal phase. They switch into what we're calling the structured signaling mode, and then they maintain that basically through the rest of the courtship. Males with higher structured signaling rates, that is the males that do this combined signal more frequently during proximal courtship, have shorter mating latencies. So they mate sooner. But heavier males had lower structured signaling rates. So it seems to be kind of maybe some sort of trade-off there. And finally, the males that were muted, the males that had their abdomens fixed so that they were, I can't say that they were totally unable to, they still tried to vibrate, so there was an attenuation of the abdominal tremulation rather than a really muting, but they took significantly longer to mate when they did. So the summary from this part of this study was basically that it's clear that males use at least 3 distinct signals during courtship. These signals do carry some information about males, and they organize these signals into a composite multi-component display during courtship, and that organization is related to mating success. So there were no effects on absolute, on whether or not males were able to mate. The significant effects were all on the latency, mating latency. That's I don't know, that's something that it's something to talk about, I guess, but I think the conditions in the lab, you put animals together in a small space, favors higher mating rates than in nature. So it's a sample size problem in that sense. Okay, so this is the end of the Black Widow story. And the next step was basically, one of the goals is to look at other species across the genus. They all have very similar signaling repertoires, but we don't know whether this composite signal occurs in other species or really anything about how the organization of signaling varies across the genus. And there's some evidence that there might be interesting patterns here. For example, the clip I showed earlier. Now I don't know if it's noticeable to you, but to me this is a very different mood for a black widow male courtship. And this is a pair from a different population from the one that we studied in the main study. And there's some genetic evidence that the species that's widely distributed, that there's actually more than one species there. So that's something that's interesting to follow up. But in the meantime, while we're trying to collect other species of Latrodectus, we do have another Theridiid in the lab, which is Steatoda nobilis, the noble false widow. So these animals are closely related to Latrodectus, but a different genus. They're an invasive species. Their native range is believed to be the Canary Islands, but they're now found in quite a few areas of the world, including North America and Europe. And this project was, some of it was done by a master's student of mine, Shagufta Raman. She was here for the Congress a couple of years ago. And now PhD student Zahir Coutinho is continuing the project. So in terms of signal repertoire, I've described the black widows, abdominal tremulation, bouncing, web plucking. False widows also have at least three obvious components in their courtship displays that, you can see definite similarities. They have an abdominal signal, a body tremulation, which is basically the equivalent of the bounce. And I apologize, I realized when I was making these slides that, the terminology, a new student came along, gave names to the things that they were observing that didn't quite match. It's really obvious that some of these things should have the same name, but anyway. And web plucking. Okay, so this is Steatoda abdominal tremulation. And what you might notice about this one is that there is a series of waveforms generated with one movement of the abdomen. So in this case, it's not a one-to-one tremulation. One movement of the abdomen generates this burst. So the lower trace there represents the waveform generated by one cycle of abdominal movement. So this is stridulation. And if you look closely at the junction between the cephalothorax and the abdomen, there is, I don't know if you can see the ridges on the cephalothorax, and then these pegs on the anterior edge of the abdomen that scrape against one another and generate a higher frequency waveform for one movement of the abdomen. The body tremulations these guys the males are a bit bigger in this species, so they're some of the signals come across, and there is a size dimorphism, but it's not nearly as extreme as in the black widows. That's a female on the edge. And then finally, plucking. So very clear correspondence between the signal components, but very, very different styles and mechanisms, you might say. In both cases, the males, when the web plucking, for example, they're using their second pair of legs to vibrate individual strands of the web, but in Steatoda, it has this consistently rhythmic quality that's not at all present in the black widows. So, are these signals organized in any way comparable to the black widows? So I've got a different representation of the behavioral sequence here. So this is just a trace showing the time of occurrence of the different behaviors that we recorded in observing these courtship interactions. And I'm sorry, but I think the labels don't perfectly line up. But the blue ones in the middle are the plucking signals. The top sort of puce color is our copulations. And then the other one to notice is the red at the bottom is walking. So you can look at the early stages of courtship in Steatota, and there's more walking. And there's also more tremulation. The next two rows above that are tremulation, bouncing, and abdominal stridulation. So there's early stages of courtship. Males are walking around a lot, bouncing on the web, and stridulating pretty much continuously. And we call this the exploratory phase, or because of the fact that the males are basically moving all over the web. And then at some point, they localize the position of the female and start focusing their activities closer to the location of the female. And we're referring to this as the courtship phase. And in this phase, the dominant signal is really plucking. So they sit in one spot and they pluck, like that clip that I showed. So there's a possible distinction between styles of plucking. Haphazard plucking is when males are doing it while walking around versus sitting in that highly rhythmic sort of pattern that we're calling sustained plucking. Okay. So there's possibly a parallel to the packaged, organized, multi-component signal in this species as well. So that's indicated here. If you look closely at male behaviors in some of these situations, there seems to be a sequence of plucking, stridulation, body tremulation, and then back to sustained plucking. So if you all right. Plucking for a while, walks around in a circle, stridulating. Bounces. So this, I think, needs more observation. It's a tentative identification of this kind of a pattern, but we haven't quantified this in the ways that we looked at the black widows yet, but possibly. Okay, at some point, after the male has directed a whole lot of web plucking and other signals towards the female, she changes posture, and this is the indication of receptivity is the female moves out of towards the male and adopts a specific posture, and then we enter the copulatory phase of the display, and this is another obvious difference with the black widows. It's much longer, so there's a much longer interaction around during copulation, and there are multiple insertions separated of varying duration, separated by periods of behavior that involves plucking and what we call lunges. So, and at some point the pair separates, and then the males may go back to something that looks like the initial exploratory part of courtship. So here's so I apologize for this, but we made web frames, and these the females will build better webs if they have a little refuge to hide under, but we made the mistake of making a refuge that would obscure part of the video. So, of course, they like to hide under there when the things happen that you want to see. So try to see through that plastic lid. The male's on the right, the female is at the left. Oh yeah. So listen to the plucking. Female changes posture. Going faster. And then he starts this lunging behavior where he's trying to initiate copulation. So this is another signal characteristic that males accelerate the rate of plucking in response to the female's receptivity signal. And then the mating interaction so they alternate palps with each copulation. And then he would go back to plucking when they separate, and those lunging movements, but with the opposite palp on the opposite side of the female. Okay, so what do we know about how any of this correlates with mating outcomes? Heavier males are more likely to mate, and they tend to have significantly longer copulation durations, the total duration of insertions. And if you quantify the relative change in plucking rate that males do after the female signals receptivity, that's correlated with copulation duration too. So males that increase their pluck rate more mate longer. But the increase in plucking rate is also negatively correlated with male weight. So it's, again, reminiscent of the pattern in black widows where maybe there's some alternative, you can be big or you can be fast, or you can compensate for being less big by greater signal effort or something like that. And we don't have any data yet on the, as I said, on the possible effects of that structured signal. Okay, so to line them up in parallel, I guess, there's three distinct signal types. I think how you divide up the signals is a question of how close you look, but there are at least three clearly distinct signal types in each of these species, with some of them correlating with male phenotype. And in the black widows, the males organize these signals into a kind of composite multi-component display. There's some evidence that that's also the case in Steatota. We don't know that for sure yet. But there's a big difference in terms of the proportion of these interactions that are directly related to insertions and repeated matings. That's extended in Steatota relative to the black widows. And there's this pattern that of males of kind of different correlates with mating success or with copulation duration, with larger males or greater signaling effort being kind of alternative, well, I don't want to say alternative strategies, but maybe a trade-off between two different parameters that correlate with mating success. Okay. Now, I must have saved to the wrong file here because I thought I had Do I still have? Yes. Have I got a couple minutes to show you? Oh, I see. Why are these hidden? Do I need to? I need to share screen again. Yes. We can leave that one up. That's okay for these ones. Okay, so I've basically been exploring a whole new world of spider signal diversity down here for the last couple of weeks. And this is very quick and rough because I just selected some segments from the oodles and oodles of raw footage that we have. So there we go. That's covering the well, this is Pauacosa. So the male is drumming on the substrate with his palps, and then when he gets more excited, I think there's another one. So this is the male at the bottom. Palps. Leg tapping. So palp, drumming, possibly stridulation, abdominal movements, and leg tapping. Okay, this is Schizocosa maliciosa. Again, the male's tapping with his palps. It actually takes a lot longer than this. Okay, this is Lagodes. Okay, this one's kind of quiet. So these ones are on webs, and they're able to move very quietly on the web. So eventually this male will So they also pull the web and displace the reflection point for the beam, so it makes a lot of noise. Okay. Again, same species. This is later on. Oh, that's the same one. Sorry. So, So there are a couple of interesting things about this one. One of them is that they pull the web, and that seems to alter the coupling between the male and the web surface. You can, in some cases, you can hear a change in the quality of the vibration as a function of the tension on the web, and they continue signaling throughout a copulation. So that's the male's abdomen. And it's actually quite loud, but it's a very low frequency signal, so it doesn't come across very well in without a subwoofer, I guess. Stop. Ooh. Damn it. Maybe I can make it. I have a power supply, but. This is the one that you were listening to, so you remember the. And finally, we have a couple of clips of the Apontia uruguayensis. The smallest ones. This male is able to time travel, apparently. Let's see. If you look closely and listen closely, you see the female's abdomen moving, and you might hear that she's responding. So there's a very consistent. And it's also the males alter the position relative to the surface, and that again has an effect on the. Okay. Okay. So I'm sorry. Maybe I should have done all highlights of the local species, but. I'm happy to answer any questions if there are any, and thank you very much.
¿Preguntas?
Thanks for your talk. Well, it was very cool. I have many questions, but I was wondering, like, if the position of the bean is important, like in the black widows, if, like, the quacking of the strings is important, like which string are you quacking or not?
Yes, that's a good question. And I don't know the answer, but not just in black widows, but especially in Steatota, where the males always use two legs, but they don't. They don't. The timing is variable, so they're not simultaneous. Sometimes they are. Sometimes they're alternating. So I think it suggests that there might be spatial information available because you have separate stimulation points that might be something that the females can assess. So we don't know, but I think it's possible.
And the only response of the female is like the changing of position, or is there any communication there?
With Steatota, it seems, as far as we know, it's just the change of position. With the black widows, the females sometimes make an abdominal pulse, that like a single boop.
And the last one is about like the sensory system in the female is like known, like any use of that.
Yes. So we know a little bit about that. The primary sensory mechanism for vibration is mechanoreceptors that are located at each joint. So they're distributed over the entire, every leg joint over the entire animal. So they have an array of receptors that are positioned around the web. And we have some evidence from black widows that there's a certain, to a certain extent, there's a frequency segregation in the sensitivity of the receptors. So the receptors more distal are more sensitive to higher frequencies, and the more proximal receptors are more sensitive to lower frequencies. So if you think of a female black widow, they're actually hanging on the web, and the web's vibrating. If it's a high frequency, the body tends to stay still and the legs move like this. And if it's a low frequency, then the body tends to oscillate. And so it stimulates different receptors differently. Additionally, the females can adopt different postures, so they can retract their legs. And if they do that, it favors some receptors over others. So if they're hungry, they may be able to tune their bodies to prey frequencies preferentially, or if they're receptive, they may be able to listen at frequencies that are characteristic of males, and there's broadly some separation of those frequencies. So it's all the peripheral sensory system that we know about, and that's about as much as.
¿Otra pregunta?
Hola. Andrew, first of all, thank you for coming to Uruguay. It was so nice having you here, and I hope this is the first of several visits.
Me too. It's been wonderful to be here.
I was wondering whether you know in the Black Widows or in Steatoda, if there are differences in male signals when the female is virgin in comparison with females that perhaps have mated lots of times with other males, or?
The short answer is we don't know that, but there are certainly differences in male effort that could correlate with that. But in terms of the parameters of the signals, I have no information on that. So only that they signal more or less in certain ways that.
Okay, and I have a second question about the species of Uruguay. In some of them, the females responded to. Can you remind me which species the females you saw a response of the female signaling the male?
There were several actually, and it's interesting.
Most of them.
Yeah, but it's not consistent. So one of the interesting things about this has been that the kind of pattern that the courtship plays out is very different. So sometimes there's successful mating with female signaling, without female signaling, with relatively, sometimes the males don't do everything that we've observed them do. It seems to be highly variable. Yeah.
Thank you so much, and for the rest, not for you and for you also, that we have been studying these animals for more than 40 years, and now we are learning new things that it's like opening a door to something, a very new and beautiful world. We are very excited with this. Thank you.
Thank you.
Thank you so much for being here and for the knowledge. If you can explain, a brief explain about the vibrometer laser. the stuff?
The laser vibrometer?
Yeah.
So the principle is that you direct the beam at a surface, and if the surface, and then the surface has to reflect a beam back into the device. It only needs a very, very small fraction of the energy, just one speckle reflection. So if the reflecting surface is moving, then the reflected beam is Doppler shifted. So there's a minute change in the wavelength of the light that is proportional to the velocity of the surface. The device compares the outgoing and returning beam and does an interference calculation and produces a measure of the instantaneous velocity of the surface. So the output from the device is a time amplitude trace of the surface velocity. Does that make sense? Yeah. So it's like a non-contact microphone for vibration in a sense. So it can be calibrated if we were careful about how we made the measurements. We could get absolute velocity measurements, and is that like, it's in the lab.
Otra pregunta.
Okay.
Sorry for the problem.
All's well that ends well. Yeah.